What you see here is a clone of the original spruce that was sequenced by scientists at SciLifeLab, and for the first time the full genome of the Norwegian spruce was characterized.
This study was really the first study that tackled the large conifer genomes, and they are notorious because they are so very much larger than just about any other organism.
I think many people thought it was a very bold move at the time to try to tackle this genome. The genome of a spruce is about 7 times the size of a human genome, so it's very large and it's very repeat rich. So it was a super hard technical challenge, and I think at the time it was also, for SciLifeLab, it was really at the early days a statement to say that we can go, at the very forefront of science and technology and beyond what can be done today. It was a good statement from SciLifeLab from the very beginning.
I was also the person who selected the actual spruce that we were going to sequence. So I went out in the middle of the winter on snowshoes and went through a large spruce plantation and decided that this is the tree that we're going to use.
The Spruce Project first way was only to sort of map out the basic genes. We didn't even know what genes were in the genome, what they did at all, how they were connected, how they were regulated. So all of this sort of basic fundamental biology, that was really the core of this first Spruce Genome Project. At the time, there was almost no genetic resources at all for spruce, and spruce is, as you know, one of the most ecologically dominating species in the northern hemisphere, and we had virtually no tools genetically to answer anything on the fundamental biology of this species. So it was of enormous importance to start a wave of new studies that came after the Spruce Project. It's been cited more than, or almost 1,500 times so far, and it's still cited more than 100 times every year. So it's been sort of paving the way for a lot of studies downstream, working on the function and the evolution of these giant conifer genomes. I think at the beginning it was also very important for SciLifeLab to show that we had collaborators all over Sweden, and so teaming up with Umeå Plant Science Center to tackle this massive project, I think was a smart move, and it also made a statement early that SciLifeLab would change and tackle research in all research fields, not only medical science, and I think that statement came through in a good way as well.
This was a groundbreaking study in the sense that it made conifer genomics not only a dream, but actually a reality. And I think following this study, there's been many more studies looking at both spruce and other conifers. And this is now more mainstream than it was maybe 10 years ago. So I think this will have really important consequences for in practical applications like spruce and conifer breeding, for instance, where we think that these tools that we have developed throughout this project will actually be very helpful.
At that time, I was acting co-director for SciLifeLab, and in that capacity, I had the immense honor to be there to help plant this beautiful and historical spruce. We were given a copy, a clone, a tiny baby spruce in a pot. And then we convinced the botanical gardens to receive it and plant it here. It wasn't very difficult because this is a special tree, as you can see. Today we think of SciLifeLab as the most important molecular life science infrastructure for research in Sweden, and it's just amazing to have been able to be a small part of that in the beginning.
You can see now we're just planning to move into the third building on the Solna site, and we have campuses all over Sweden. So I mean, SciLifeLab has grown enormously since that time. It was basically, compared to now, a very small clique of people with a grand vision.
We thought that it would be a lovely thing to have this spruce grow here in the botanical gardens in Uppsala as a signature plant, showcasing the capacities of scientists at SciLifeLab, because cloning the spruce genome and sequencing it was not an easy task. And here in the botanical gardens in Uppsala, plants from all over Sweden and other parts of the world have been collected for centuries. It is also a place where students come to learn about the plants, and it is one of the landmarks of Uppsala, actually, with many thousand tourists coming through here every year. So I'm very proud that we have the SciLifeLab spruce as one of the signature trees of the botanical gardens in Uppsala. To be honest, you might not think of it as the most beautiful Christmas tree like spruce, but let's think about it as a scanty teenager who has not yet quite found its form. But it is a very valuable tree, and we hope it will remain here for many years to come.
Since a few years back, we're now working on an updated version of the spruce genome and a completely new genome project on pine as well. Since technology has moved on, this is now, depending on how you see it, we're talking about that we are 10,000 times better than the old study. And so the resolution and what we can see now in terms of how the genome is organized and regulated and how it has evolved is just mind-blowing. And it will really allow people to take conifers into a genomic era that we maybe wished for 10 years ago, but was not really possible at the time.